In addition to passive transport, frequently a cell uses the bond energy of ATP to move solutes across the plasma membrane. This process is called active transport. There are numerous instances in which the cell must expend the energy it has produced in its mitochondria to accomplish a life-sustaining task, obtain a valuable nutrient, or export a product. For example, sometimes cells must move solutes across the plasma membrane against their concentration gradient. This is analogous to pushing a car uphill. In this series, we will study the primary types of active transport: primary, secondary, and vesicular. There are two major mechanisms of active membrane transport: active transport and vesicular transport. Our key component is ATP. Hydrolysis of high-energy phosphate bonds transfers energy to a protein. Proteins are the working molecules of the cell. This energized protein allows the cell to import or export specific molecules regardless of their intra- or extracellular concentration. In this type of active transport, carrier proteins are required. Sometimes the cell requires materials that are too large to pass through the channels and membranes. In this case, ATP is required for a combination of processes leading to the formation and fusion of vesicles containing large particles, macromolecules, and fluids. The main examples of active transport that we will discuss in this lecture are primary active transport, involving ion or solute pumps that specifically transport the cations sodium, potassium, calcium, and magnesium, or some anions chloride, iodide across cell membrane. Secondary active transport, and exo and endocytosis. Both primary and secondary active transport involve carrier proteins. In primary active transport, the energy to do work comes directly from hydrolysis of ATP. In secondary active transport, the actual transport is driven indirectly by energy stored in ionic gradient created by the operation of primary active transport pumps. Secondary active transport systems are all coupled in that they move more than one substance at a time. If the carrier transports two substances simultaneously in the same direction, either both into or out of the cell, the system is called a symport, same or together, port for transport. If the carrier transports two different substances in opposite directions, the system is called an antiport. The most thoroughly studied example of a primary active transport system is the operation of the sodium-potassium pump. The carrier protein is an enzyme called Na plus K plus ATPase. The pump exchanges 3 intracellular sodium ions for 2 extracellular potassium ions for each ATP hydrolyzed. The more sodium ions in the cytoplasm, the faster the pump works as long as ATP is available. The ATPase may use up 40% of the ATP produced by a resting cell. In living cells, homeostasis or ion steady state is not reaching equilibrium between either sodium or potassium on both sides of the plasma membrane. Distribution of ions across cell membranes is at true equilibrium only in dead cells. In living cells, there is a great disparity between the concentration of sodium on the outside versus the inside of the plasma membrane, and the same is true for potassium. K plus is 10 to 30 times more concentrated in the cytosol than in the extracellular fluid. Intracellular concentrations of free Na plus and Cl are 0.1 or less than the ECF. These ionic concentration differences are essential for all cells to be able to respond to stimuli, store energy, and to maintain their normal fluid volume. The concentration differences are even more pronounced in excitable cells, muscle and nerve cells, because this is how these cells generate and or propagate electrical impulses, which is how they send messages or contract, as the case may be. To best understand how this pump works, go to Mastering A and P and watch the animations of the pump. You can also refer to the animations in PhysioX or iPhysiology. The diagram above shows each step in sequence. Notice that there are more sodium ions yellow outside the cell than inside, and that there are more potassium ions green inside the cell than outside. 3 sodium ions in the cytosol bind to their receptor sites on the ATPase. Binding of the sodium promotes phosphorylation of the enzyme by ATP. The phosphorylation causes the pump protein to change shape, which expels the sodium to the outside of the cell. 2 potassium ions in the extracellular fluid bind to their receptor sites on the enzyme, which triggers release of the phosphate. This causes the ATPase to return to its original shape, and potassium is released into the cytosol. The pump is then ready to bind sodium again and repeat the process. The more sodium in the extracellular fluid, the faster the pump works, as long as ATP is available. Another example of an ion pump includes the calcium pumps that actively sequester ionic calcium from the intracellular fluid into specific membrane bond organelles like the sarcoplasmic reticulum in muscle cells, for example. In secondary active transport, the transport mechanism itself does not need energy. However, energy is ultimately expended to maintain ionic homeostasis. What happens is that the carrier protein moves a solute down its concentration gradient and can move another solute simultaneously without regard to its concentration gradient. In other words, the concentration gradient for one solute provides the force. The second solute gets a free ride. Let's look specifically at one of the most important secondary active transport systems for the cell, sodium-dependent glucose transport. In this case, the concentration gradient of sodium provides the main driving force for secondary active transport. A carrier protein will co-transport glucose into a cell, but only after it first binds to sodium. Both sodium moving down its concentration gradient and glucose regardless of its concentration gradient are transported into the cell. The cell must now expend energy by hydrolyzing ATP in order to pump out excessive sodium ions via the sodium-potassium pump. In vesicular transport mechanisms, materials are moved into and out of the cell via membrane-bound sacs called vesicles. Vesicular transport is used by the cell when bulk transport of large volumes of fluid and solutes is needed, or to transport specific types of large molecules. There are 2 broad categories of vesicular transport. Endocytosis is the process by which materials are brought into the cell. There are 3 major subdivisions of endocytosis. 1, Pinocytosis, also known as Phagocytosis, is cell drinking. 2, Phagocytosis is cell eating. 3, and Receptor-Mediated Endocytosis. Exocytosis includes secretion and excretion of the elimination of degraded or indigestible material. The picture in the slide above is an electron micrograph of a secretory vesicle releasing its product. The magnification is 100,000x. Pinocytosis brings extracellular fluid into the cell. No receptors are involved, so pinocytosis is not a specific process per se. However, the cell can control it. The plasma membrane forms a groove or pocket called a caveola, which surrounds a large volume of fluid. Eventually, the vesicle pinches off the plasma membrane, forming an internal vesicle called an endosome, or more specifically, a pinosome. Phagocytosis is a general term to describe cell eating. It is a mechanism by which cells can bring in large amounts of solid material. Many types of single-celled organisms use phagocytosis to ingest other smaller organisms, such as bacteria, that they need for their energy requirements. Some of the cells in our body are specialized in phagocytosis. Their job is to ingest and destroy foreign material, be it organic or inorganic debris. Phagocytic cells have the ability to mobilize the outer rim of cytoplasm, creating pseudopodia or false feet. The pseudopodia surround the material and eventually engulf it. The material is now surrounded by a vesicle made of plasma membrane and is now called a phagosome. Lysosomes fuse with the phagosome and release powerful degradative enzymes that break down the ingested object. This is the way the dust cells found in the lung alveoli keep the respiratory surfaces clear and clean of particulates that we breathe in daily. Receptor-mediated endocytosis (RME) is a selective process by which materials to be transported into the cell bind to plasma membrane receptors specific for that material that are localized to a small area of the plasma membrane. In general, the specific or target molecules are referred to as ligands. RME can transport ligands into the cell against steep concentration gradients. The receptors bound to their ligands form clusters on the plasma membrane in specific areas that have a coating of specialized proteins such as clathrin. When the material is engulfed, the endosomes are referred to as coated vesicles. When lysosomes fuse with the coated vesicles, the clathrin coating is removed. The receptors release their ligands, and the receptors are recycled back to the plasma membrane where they are ready to bind another ligand of the same type that the cell needs. The picture in the slide above compares the three types of vesicular transport. On the left is a diagram illustrating each process. On the right are electron micrographs of actual cells involved in each process. Exocytosis is the process by which material is transported out of the cell. In the example above, the cell has engulfed a bacterium which was neutralized and degraded when a lysosome fused with a phagosome. Some of the material can be exocytosed out of the cell, where it may be excreted by the body. Secretion is a special type of exocytosis. In secretion, proteins such as mucus, hormones, or digestive enzymes that are synthesized by the cell are moved to the plasma membrane in secretory vesicles. The vesicles fuse with the plasma membrane, and the contents are released into the blood hormones or into a duct to be carried elsewhere in the body where they are required. When the cell is stimulated to secrete its product, the proteins are synthesized in the rough endoplasmic reticulum (RER) and transported to the Golgi apparatus for further modification and concentration. The vesicles containing the protein pinch off the Golgi apparatus and fuse with the plasma membrane, releasing the contents. The diagram above shows the specifics of the process that occurs when the secretory vesicle approaches the plasma membrane. Notice that there are proteins called snares that are involved with the fusion process. SNARE is an acronym derived from soluble attachment protein receptor. SNARE proteins are a large family consisting of over 60 known members. The snares are divided into two categories: vesicle or V snares, which are incorporated into the membranes of transport vesicles during budding, and target or T snares, which are located in the membranes of target compartments. The fusion of the two snares docks the transport vesicle and opens up a port to allow release of the vesicle's contents to the cell exterior. We previously saw that selective permeability of plasma membranes can lead to osmotic flow and establishment of concentration gradients across cell membranes. Another result of selective permeability is the generation of a membrane potential or voltage across the membrane. A voltage is electrical potential energy resulting from the separation of oppositely charged particles. In cells, the oppositely charged particles are ions, and the barrier that keeps them apart is the plasma membrane. All living cells expend chemical energy to maintain the transmembrane concentrations of solutes far away from equilibrium. But why? It turns out that the inside of the plasma membrane has a slightly negative charge compared to the outside. This is due to the distribution of charged ions. There are more positively charged ions on the outside of the cell and more negatively charged ions, especially proteins, on the inside the cell. The oppositely charged ions are kept away from each other by the plasma membrane. This potential difference represents stored energy, which can be used for the cell to do work, and is called the transmembrane potential. When a cell is at rest, which means that it is not responding to various stimuli, the transmembrane potential is called the resting membrane potential. The cell membrane and resting membrane potential are analogous to a battery. Both a battery and a plasma membrane have a positive end and a negative end. By keeping the positive and negative charges separate, both the battery and cell membrane are able to store potential energy. In the case of a battery, when you turn on the switch of a flashlight, the polarity reverses, and this creates the power that turns on the light bulb. In a cell membrane, when the polarity reverses in response to a stimulus, the cell uses the stored energy to do work. The selectively permeable membrane is responsible for generating a voltage across the membrane because it selects what ions can enter or leave the cell. The resting membrane potential of cells ranges from -50 to -100 millivolts MV. The cells are electrically polarized. The resting membrane potential is determined mainly by the concentration gradient of potassium and the differential permeability of the plasma membrane to potassium. Potassium and protein anions predominate in the cytosol. Sodium predominates in the extracellular fluid ECF. Potassium ions diffuse down their concentration gradient and out of the cell through channel proteins called potassium leakage channels. The loss of potassium results in a negative charge on the inside of the plasma membrane, mainly because of the presence of negatively charged protein anions. In addition, potassium ions also move into the cell because they are attracted to the negative charge established on the inner plasma membrane. Therefore, a negative membrane potential is established when the movement of potassium ions out of the cell equals the movement of potassium into the cell. Flip-flop in the transmembrane potential is behind muscle contraction, secretion of glands, and transmission of information in the nervous system via action potentials. The picture on the right shows a segment of an axon. Notice both the charge differential as well as the differences in the concentration between the various ions inside and outside the membrane. The resting membrane potential is maintained by the sodium-potassium exchange pump, which uses ATP to return the relative concentrations of sodium and potassium across the cell membrane in a steady state of disequilibrium. How do we know this? Many times the way physiologists figure out what something does is to break it and see what goes wrong. This is true of the sodium-potassium pump. It was known for a long time that tribal hunters would use poison arrows to paralyze their prey. When physiologists investigated what was causing the paralytic action of the poison, they found that an extract from the seed of certain African plants called Ouabain, pronounced Ouabain, stopped the activity of the sodium-potassium exchange pump. What happens is that when Ouabain is present, it sits in the binding sites for potassium on the pump. This effectively inactivates the pump, and sodium and potassium diffuse down their respective concentration gradients until they reach equilibrium across the plasma membrane. This brings the resting potential to 0 MV, and therefore the membrane no longer is able to respond to stimuli such as signals for muscle contraction or nerve conduction. There is no stored potential energy in the membrane.